Hello everyone, and welcome to today's webinar, DeepCLIP Predicting the Effect of Mutations on Protein RNA Binding with Deep Learning. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Thomas Doktor, molecular biologist, Department of Biochemistry and Molecular Biology, University of Southern Denmark. Thomas, you may now begin your presentation.
Okay, welcome, and thank you for joining this webinar where I'm going to present our tool called DeepCLIP and ways of using it to analyze and predict the effect of mutations that might affect protein RNA binding. So starting right from the beginning, the central dogma of molecular biology, we produce proteins from DNA via RNA. And of course, RNA is not just a naked RNA as in this picture, but it is often in a complex with a lot of RNA binding proteins. So if you look at this figure, for instance, we can see that RNA is transcribed from DNA and then undergoes a lot of processing and binding a lot of these RNA binding proteins. Some of them are generally binding to RNA and others have more specific sequences that they bind. If you go right to the top for looking at transcription, we can see that there's a lot of proteins involved in that. In our group, we focus on splicing, and splicing is the removal of introns and the ligation of exons into a mature mRNA that also contains a 5' cap and a 3' poly A. So that process is regulated by a number of different factors. Of course, the spliceosome is this multi-megadalton complex of many different molecules, both proteins and RNAs. And specifically, the use nerves recognize these splice sites that specifically delineate the beginning and end of introns and exons. But the regulation of the use of these exons, whether they're included in the transcript or not, is often regulated by splicing factors, which are RNA binding proteins. And they bind to these enhancer elements and silencing elements. And if they're in exons, we call them exonic splicing enhancers, for instance, for an enhancer, and an exonic splicing silencer for a silencer. And the balance between those is tightly regulating whether these exons are included or not, and sometimes it's a very delicate balance. And of course, this allows the cell to produce multiple different variants of the same protein via different transcripts. So if we're looking at an exon here, we can see that it might be included in some cases, or it might be skipped. There can also be mutually exclusive exons, or there can be alternative splice sites, or simply a retention of an intron. So in normal cells or in normal conditions, the exon might be included and producing a normal transcript that results in a normal protein. But if there is a mutation that might affect these enhancer elements and affect the binding to their respective RNA binding proteins, that could result in a misproduction of a transcript that does not contain the exon. And that can result in dysfunctional proteins or degradation of the transcript via NMD. So those mutations that may not even disrupt the reading frame and may not change the amino acids, because they disrupt splicing, they can have a potentially strong deleterious effect and cause disease. We can also have mutations in the introns, and sometimes they can activate what is known as a pseudoexon or a cryptic exon that is part of the intron which has a very high similarity to an exon, but is not normally included. We might have a mutation that then creates or destroys an RNA binding motif, RNA binding protein motif and that results in activation of the pseudoexon that might be included in this transcript, disrupting the normal function. So if we go back here and view in this slide here, you can see that there's a number of different elements that all affect splicing of an exon. So in all of these motifs that bind RNA binding proteins, we have fairly motif specific proteins that recognize specific sites. And mutations in these, they can disrupt the binding, or so either by increasing it or decreasing it to a kind of aberrant function, essentially. But RNA binding proteins, they're also depending a lot on the context. So some of these motifs might be in close proximity to each other, and so they're also in competition with each other. So they're highly dynamic, and simply having a motif does not necessarily mean that this is a functional binding site. And that is a complication in predicting whether a motif binds to a particular protein or not. If we go in and look what is the normal variation in the human genome, then from the 1,000 Genomes Project, we know that there are tens of thousands of variants in exons, and many of them are synonymous, so they don't really change the amino acids, but all of them might change splicing. We also have more than 1.5 million of these intronic variants on average, and essentially every one of them has some small chance of activating or just disrupting splicing. So all of these are really a part of what makes us different. So they can cause variation, but they can also cause disease. And of course, there are so many, we cannot functionally test everyone. So we do need in silico tools. These mutations and variations, they also act together. So this is a case of a disease mutation here at position 362 in the ACAT-M gene, where a patient mutation changing the C, the natural C to a T, disrupts enhancer element and causes exon skipping. So here we've used an amino gene and introduced or ordered these mutations so that we can test their function. So if you look in this lower picture here, we've done transfected cells with the amino gene and purified RNA and done an RT-PCR. So and then separated on an agarose gel. So we can separate the long band, which contains the exons, from the exon from the lower band, which does not. So in the first lane, we have the wild type, where we see inclusion of the exon and also some skipping. This is sometimes the case in mini genes, that you have a little bit different splicing from the wild type. Then when we have the disease variant 362T, we get predominant skipping and almost no inclusion. And actually, this is what causes the phenotype, the disease phenotype in this case. There is an amino acid change, but it doesn't have a dramatic effect on the enzyme activity. So it is the skipping of the exon that causes the enzyme deficiency. But there is also a normal polymorphism, a normal SNP, located at position 351. So normally there is an A, and in this case, the SNP is a C. And this is a completely silent mutation or variant that does not change the amino acid, and it also does not change splicing. But when you have both the normal, you have the variant in the C, then if you have a introduction of the disease causing a variation, you actually have protection from the C here. So the ultimate outcome of these SNPs are depending on which context they're sitting in. So we cannot just analyze each of them individually. So this just creates an even bigger problem of understanding the particular functions of these SNPs, because then we have a number of different variations. So having in silico predictions tools to analyze these are really essential. So this is not a new problem, of course, but it's a difficult problem. In the past, we've used position weight matrices such as ESE Finder, where we can analyze the motif directly. And that means that we're looking at each base individually. So in this motif, the bases are independent from each other. So having a single base change only affects that particular base. So there's no context modeling. Another method is to simply have database lookups. So we know some of those motifs, or we have some statistical modeling of motifs that might be enriched in exons versus introns. And these rescue ESE and FastESS are examples of that. In more recent years, there's also been machine learning methods such as support vector machines. And one of those is GraphProt, which introduced the modeling of structure as well. So GraphProt has a step where you input very large, long sequences and spend a long time modeling the structure, and then that structure is part of what is giving the ultimate prediction in the end. There's also neural networks. DeepBind was the first, and DeepBind works by having many different motifs for essentially for the same protein. So instead of having just one position weight matrices, it has a number of different motifs and features that are recognized. Another case or example is IDeepS, which works a little bit like DeepBind, and it recognizes motifs, but it also has a structure modeling step with a different tool similar to GraphProt. So you input both your sequence, and then you need to run a structure modeling and use that as an input as well. So these existing tools are all in some way lacking in features when we were using them. In particular, the context modeling is focused on structure. And many RNA binding proteins are depending on certain structures, but that's not the only type of context that could affect binding. It could be that they act in concert, so that having multiple motifs or having different types of protein binding and having a synergy is actually increasing that binding strength. It could also be that some proteins bind specifically in near 5' splice sites, so that would be a type of context that was important. Another problem with having structure prediction is that it requires long sequences. So if your model is depending on this structure, you really don't have as high efficiency when you're predicting on shorter structure, where your structure modeling doesn't really work. And then the structure prediction is itself an in silico step. So it's not error-proof, and it's noisy. So you're also introducing noise in some way into the system. And there is also generally a lack of individual base scores. So in the motif, if you have a position weight matrix, it typically has a length of 6 or 7 or 8. Then you get a score for those 8 bases. And you don't really know if you change a base. It might change that particular motif, but it might also result in that you have a little bit of shift of a motif. So you need to do a lot of different comparison and scoring with these position weight matrices. So that's one way that you can kind of get individual bases, but it takes a long time and it's not straightforward. And if you have these machine learning models and neural networks, you often get just a single prediction score. So you can actually then introduce a mutation on each base, all possible mutations. But of course, that greatly increases the number of predictions that you need to run, and you also need to manually analyze them, or at least have the capability of doing that. And there's also not a support of variable length input for all of these tools. So some of them require fixed length input, and some of them do allow variable length input. But we sort of lack a tool that combines all of these. So that's why we designed DeepCLIP. So we designed it to have arbitrary context modeling, where we don't really tell the models which kind of context that we're looking for, but we have a model or a design of the neural network that allows a certain level of context modeling. And that also means that we don't need any extra tools to predict structure. So we just need the sequence. And we introduced a binding profile so that the overall result of the analysis is not just a single score, but it's an individual base score as a binding profile. And because we have these binding profiles, we can also provide variable length input, and variable length, it does affect the overall prediction score, but I'll show later how the binding profile actually allows us to still examine quite fairly different lengths of input. So DeepCLIP is a neural network, and just briefly to explain neural networks, they are these nodes in different layers that are all interconnected. So usually we have an input layer, and all of those inputs are then fed into the network, where there is a number of different hidden layers that we don't really see, and they are connected so that they get input from the previous layer, and then they output something to the next layer. And ultimately, we have an output layer that can be many nodes, but it can also just be a single node with a range in values that you're interested in. So this is a very general approach. So as you can see here on the left, the prototype multi-layer perceptron, this is this kind of network with just a single hidden layer. In DeepCLIP, we also use something called convolutional neural networks, and they have fewer connections, so that the nodes in the convolutional layer, they are only connected to some of the inputs. So for instance, here, each node is connected to 2 of the inputs, sort of in a sliding window. So we move one step forward, so the next node is connected to the next 2 nodes of the input. And all of these nodes under the convolutional layer, they share parameters. So what really happens is that here we can identify local features and local patterns. So this is how we extract motifs of the RNA binding profiles, or the RNA binding proteins. We also use a type of network that's called a recurrent neural network. And what's unique about this is that not only are the nodes connected to the input, but they're also connected to themselves and the other nodes in the layer. We then use a different type or variation called long short-term memory that has multiple parameters inside the nodes that allow it to be activated after certain patterns. So they can forget things, and they can just choose when to become activated. So it resembles in some ways a type of memory. So when we combine all these things, we have the overall DeepCLIP architecture where we have input. We then encode it into a numeric string so that we can process this with these equations that a neural network essentially is. We have then the convolutional layers, and then we use a type of enhancement called the winner takes all. So we have a number of different sizes of these convolutions, so different filter sizes, different motif sizes essentially. And for each of them, we enhance just one part of the sequence. We then combine all of the different enhanced sequences with the original sequence, and then we feed that into our LSTM layer. So this long short-term memory layer where we are able to model context. We then use something called a bidirectional layer, but it's really just 2 different LSTM layers where one layer is fed the input from the forward direction and the other layer is fed the input from the reverse. And that's because these memories, they only work, they can remember the past, but they can't predict the future. So they remember what the sequence was before. So in order to get context modeling in both directions, we need to feed in the sequence in both directions. After that, we then combine the 2, and then we sum them into a binding profile. And that is the primary result of DeepCLIP. We can then squash with an output layer this binding profile into a single prediction score. And that's what we use when we train. Then we only use the prediction score and not the binding profile. So once we've trained a model, we can then extract the motifs from the convolutional layers. And here you can see that we have some UAG binding protein. So it does really seem to recognize some patterns in the data. When we run a prediction, we can then extract the binding profile, and we can extract the overall prediction score. So here you can see that the binding profile, it has this wavy or curvy pattern where it sort of highlights motif or sequence patterns that are sort of contributing positively or negatively to the prediction score. So if there is a lot of negative scores, then it's producing a low prediction score. If there's a lot of positive, it's producing a high. And we call it a binding profile, but it's also a profile of which areas of the sequence seem to have a positive contribution to binding. So we call this DeepCLIP because we're using deep learning with several layers in the neural networks, and then we're using CLIP data to train the model. So CLIP is cross-linking and immunoprecipitation, and it's a number of different techniques with variations on the same principle, where you can identify specific location of protein binding in the genome. So in this experiment, we irradiate cells with UV light, and that induces cross-linking of the RNA binding protein to the RNA, preferentially at the site of binding, or hopefully. We then degrade the RNA, so we get fragments of RNA that are shorter and ideally only bind one protein. We can then use a specific antibody to immunoprecipitate the complexes with a specific protein of interest and their cross-linked RNA. So then we can purify these complexes and sequence them on these novel, massively parallel sequencing instruments. And then all of those sequences we can map to the genome and then identify its binding site by looking at where there is a high coverage. So for DeepCLIP, once we have these binding sites, we then regard them as positive binding sites. Of course, there will be false positive in this data set, and there will also be many positive binding sites that were not picked up on the experiment. But for simplification purposes, we assign that as a positive binding site. Then in order to have a suitable negative background, we could either shuffle that sequence or just pick a sequence at random. But we've selected to shuffle the peaks that are in the gene and then pick the same size distribution peaks in the same gene. So essentially for each peak in a gene, we move it to a random location within the same gene. We then check that it doesn't overlap any of the other peaks that we're using in that, both positive and negative, and then we extract that sequence. So that we define that as a negative background. And that hopefully produces a background that is as realistic as possible. Of course, we might have false negatives. We might have a random location where this protein also binds, but in most cases, it works fairly well. So once we have these two, we can input them into DeepCLIP, and DeepCLIP will then partition them into a training data set, and a validation data set, and a test data set. So in this way, during training, we can validate if we are doing well. And then finally, when we're done training, we can test the model on this final test set. So if we want to compare DeepCLIP to other tools, the best way is to do this tenfold cross-validation, where we simply repeat this 10 times. And then we make sure that each of these 10% partitions is used exactly once as a test set, exactly once as a validation, and then 8 times as a training. So in that way, we have 10 models where each model has not seen 10% of the data. So we can have a prediction on the full data set without having tests input that was already seen by the model. Because whenever you're training a model, if you predict on the same type of data, it will have a very high score without being as useful in real world. We then measure this performance with something called an area under the ROC curve, the receiver operator curve. And the idea here is that if you get two sequences, one is a negative, one is a positive, what is your chance of guessing correctly? So if we look at the area under this curve, we have a 90 score, or 0.9 if you're measuring just from 0 to 1, and that means that we have a 90% chance of guessing correctly. And that's good. If in the perfect world, this classifier would be completely up in the left corner, and it would have an area under the curve of a 100 or 1, depending on how you're seeing it. So if you have a very bad model, it will follow this dotted line in the middle, and that just means that the model is no better than flipping a coin. So we want to be up in the upper left corner. So in order to compare DeepCLIP to the existing tools, we use this benchmark data set introduced by the GraphProt paper, where there's a number of different CLIP sets where all of these tools have already produced data of this kind. So we then compared DeepCLIP's performance, and we compared it both to GraphProt, which does have a binding profile, but it's more something that you can produce with the model in addition to the prediction score. So the prediction score is not itself derived directly from the binding profile, but it does produce a binding profile. But when we compare DeepCLIP with GraphProt, we can see that we have a significant improvement in the performance. So in most cases, DeepCLIP will provide a better prediction and correlation than GraphProt. If we compare with other tools that do not have binding profiles, that just have a single score, some of them are improved compared to GraphProt, but in this benchmark, we can see that for each of them, DeepCLIP has sometimes a small but statistically significant higher performance. If we look at the kind of biases that affect these, for instance, we can see that there are multiple different CLIP methods. So there's HITS-CLIP, I-CLIP, and PAR-CLIP. So does that method itself affect the model performance? And at least from this data set, there is not a statistically significantly different difference between the methods, but some of them are quite small. They're only a handful of HITS-CLIP and I-CLIP data sets, and most of them are PAR-CLIP. If we look at the number of inputs, there's only a very weak and non-significant correlation. You might assume that the more input that you have, the better the model will be, but more input also might not be more stringent input. So there might be the same level of noise in the system. Input length also does not affect, but if it does, it's a weak correlation of sorts. If we look at the sequence content, then we can see that there is a statistically significant negative correlation with the GC percentage and positive for the U fraction, the U percentage. And that's probably because CLIP experiments are known to be affected by this bias because U cross-links more efficiently. So there is a tendency that if you do find a binding site, then that motif will be very close to a bunch of U's, and those were the U's that cross-linked. So the training data is biased in that regard. If we look at the other bases, A, C, and G, we can see that at least G is also negatively. So we have fewer G's, and so they have been the high U fraction is at the cost of a lower G. So does that affect models? And yes, some models seem to be more. They seem to recognize a CLIP site as much as they recognize the protein site. So for instance, here we have the POM2 model, and when we use the positive POM2 and then the positive from a different data set, the Quaker, we can see that, yes, the POM2 model does predict high scores for the positive binding sites, but there is also a tendency to predict high scores for the Quaker sites. Whereas if we use the Quaker model, trained on Quaker and negative background for Quaker, we can see that it separates these two POM2 and Quaker data sets quite nicely. So what if we look at the motifs that these models found? Then we can see that the POM2 model actually identifies the known POM2 motif, just like the Quaker model identifies the known Quaker motif. But they also both have these CAU-rich motifs that is part of the model. So that might be a PAR-CLIP signature, or in some manner that is affecting the POM2 model more. So is this is a problem not just for DeepCLIP, but also the other tools, because this is a bias in the training data, and it's sort of incorporated into that benchmark data set. But in DeepCLIP, we also have the binding profiles, and the binding profiles contain much more information because it's not just a single score. So in DeepCLIP, we allow people to produce binding profiles in what we call a long prediction mode, where we simply have a sliding window and then take the central binding profile score of each nucleotide. So then we can produce this binding profile across this transcript, JMJ D1C transcript, which is almost 9 kb, so 9,000 base pair long. And of course, having such a long binding profile, it kind of looks like a noise recording with a lot of noise in. So we then identify binding sites for both POM2 and Quaker as having this sliding window where at least 9 nucleotides must have at least an average score on the binding profile greater than 0.3. So we can then map to this transcript, which is known to bind both Quaker and POM2 sites. So in the transcript structure in the cartoon, we have in a line we have the 5' UTR, and then in the box we have the reading frame, and then in the line again we have the 3' UTR. On top of that, we've mapped the experimentally identified binding sites, Quaker sites in blue and POM2 sites in red. And then we predicted using the binding profiles and this sliding window algorithm likely binding sites for both POM2 and Quaker. So you can see here on the PAR-CLIP, we have a number of POM2 predicted binding sites from the DeepCLIP model based on this graph prod data set, and several of them, they overlap these experimentally validated binding sites, and none of them overlap any of the Quaker sites. On the other hand, the Quaker model has predicted binding sites where one of them is overlapping the experimentally identified, and one of them is very close to. We also made POM2 models and Quaker models from the ENCODE dataset eCLIP from the K562 cell line. And here we got more POM2 prediction or binding sites, but they also tend to be mostly in the 3' UTR and overlap with the known binding sites. So even when we have these biases in the training data that do really affect the overall binding or overall prediction score, by having these binding profiles, we can actually mitigate some of that bias. So the models are useful even when they are biased. Okay, so on the benchmark data set, we didn't have our own provided backgrounds. So in order to test if this way of constructing the background is also useful, we created these new models of HNRNPA1 and SRSF1. And HNRNPA1 is a classic inhibitor of splicing and inhibits inclusion of exons, whereas SRSF1 is a classic splicing activator and promotes inclusion. So those are often involved in regulating splicing, and there are many cases of mutations affecting binding of these that cause exon skipping. So here we have the motifs of HNRNPA1 at the top, and also the ROC curve. So you can see that we have fairly robust performance of this model across the different data sets. We then pick out the best performing one and produce the motifs from that and use that. And similar for SRSF1, where we used an eCLIP data set from the ENCODE data consortium, we also have this fairly robust. So we then use these models to examine this curated data set of 37 exonic mutations that cause exon skipping and 45 exonic mutations that do not cause skipping. So they're known to be neutral. And then we looked at, with a 15 nucleotide input, so the variant in the middle, and then 7 nucleotides in each direction. We then looked at the change in score from the wild type to the variant and analyzed in the skipping group versus the non-skipping. And for A1, we see maybe a little tendency to an increase in the skipping group, but not a statistically significant. And that's probably because there's many of these that are regulated by multiple proteins, so it might not necessarily be A1 directly that is causing the exon skipping. But for SRSF1, we do see a significantly decrease in score that is greater for the skipping mutation than for the non-skipping. And SRSF1 is an SR protein, and many SR proteins have fairly overlapping binding affinity and can substitute for each other. So this may be indicates that both that SRSF1 is often involved, and so that a mutation that causes exon skipping might be disrupting an SRSF1 binding site, but also might indicate just in general that binding of SR proteins is affected by these skipping mutations and not so much by the non-skipping. We can then combine them, so it could be an interaction, so it's a combination of both gaining A1 binding and losing SRSF1. So we take the change in the SRSF1 score, and then we subtract the change in A1 score from that, because A1 is a negative factor. And then we still have this significant change or difference between the non-skipping and skipping. And if we have this if we apply a threshold in the combined score of about minus 0.4, we can see that we have many more of the skipping mutations that are below that, and just a single one in the non-skipping. So applying these might actually be allowing in some way to handpick or at least prioritize mutations in exons that are more likely to also cause exon skipping. Of course, that also depends on the exon itself. We also try to use a 75 nucleotide input, and here you can see that we see some of the same trends, but the range in scores is a lot smaller. When we combine them, the p-value is lower actually than either in separately, but it's still not at that statistically significant level, but we still see that trend. So obviously the length of the input affects these scores. And if we look at one of them, the ATP7A exon 20 mutation, this is located in the middle here. So here we have the DeepCLIP binding profiles, where we have the reference in black and the variant in red. And we can see in the binding profile that there is a clear increase in A1. So this is with the A1 model. So there is a clear increase in the A1 binding predicted by the binding profile. But if we look at the overall score, it's going from something close to 0 to something just a little bit higher than 0. So not something that has a dramatic change in terms of absolute value, but this binding profile has a lot of negative values if you look in the y-axis. That's why the context in general might be negative. So the model itself is trained to recognize A1 CLIP sites. So maybe this would be a poor CLIP site, but it still might be an A1 binding site according to the binding profile. And if we then just use the 15 nucleotide input, then we get we still get that same binding profile pattern where the A mutation increases the binding profile values, but we also get a more dramatic change in the overall prediction scores, and these scores range from 0 to 1. So here we have something that is below the mean or below the midpoint of 0.5 to something that is above it. So of course these patterns will affect other tools that are looking for different motives as well. But having this binding profile really allows us to sort of, even though we have very low score, we can still see the effect of the mutation. So this was on sort of real endogenous data where we have known mutations that have been known and shown in humans to cause exon skipping. But we also wanted to see whether in vitro experiments could correlate with these DeepCLIP predictions. So we performed a surface plasmon resonance imaging, which is a technique where you have a gold-plated plate, where the oligos can be covalently attached. And then you have flow of proteins, and depending on how much they bind, we can measure that difference. And we do that by shining a light and then measuring the angle of dispersion. So if there is some binding to the oligo, there will be some dispersion. If there's no binding, it will have a different angle. And if there's a lot of binding, it will be even more different. So by measuring this angle, we can estimate or quantitate how much is binding to these oligos. So the way that the experiment is run is that you have your oligos on the plate without any proteins. Then you start injecting a solution with the purified recombinant protein. So there's only this particular protein. And then you do that for for a period of time, and hopefully it will reach steady state. And then you start washing away the protein to dissociate. So then you can get fairly complicated modeling of the kinetics of this protein binding. So in this experiment, again, using the ATP7A exon 20 example, here we use this 15 nucleotide sequence as an oligo. and then measure the amount of recombinant A1 binding. So here in the wild type, we then have these different concentration of the protein in different runs. So we run them in duplicates, and then we model that in a single model that accounts for differences in concentration. And then we plot that on top. We then take the maximum of that model here on the response curve, and then compare that in the variant. And as you can see here, there is a greatly increased A1 binding to this oligo, as predicted by the binding profiles of both the short sequence and also the long. So if we do that for a number of these exonic mutations, where we have both the wild type and the variant, and do it for both A1 and SRSF1, we can then compare the DeepCLIP prediction scores with the maximum of this model fit. And as you can see, there is a very linear and statistically significant correlation between DeepCLIP prediction scores and then the actual measured amount of protein binding in this in vitro setup. So these models that are trained on this in vivo data, they still have a good correlation with in vitro experiments. We also used, again, the minigene with our favorite minigene, ACAT-M. So looking again on this patient mutation at precision 362, where we have a T, we then ordered new mutations surrounding that, so that for these three base pairs, we had every possible combination. We then transfected these minigenes and purified RNA and performed RT-PCR on banana agarose gel, so we can then visualize here on the gel and also do a semi-quantitative quantitation of the percent spliced in of the exons, so the inclusion levels. So you can see here that we have ranges from almost 100% inclusion to less than 10% inclusion in the patient mutation. So there's a good range of scores. So then we again produce DeepCLIP scores of these mutations across this site and measure and then compare to the inclusion levels. And again here for the A1, we see again confirming A1 H9P1 as a splicing inhibitor. So the higher the DeepCLIP score, the lower the inclusion level. Whereas for SF1, we see a positive correlation. And when we combine them, we get perhaps an even better correlation than just, for instance, SRSF1 alone. And we know already that for this exon, it's often a combination of the two. If we just plot the amount of change relative to the wild type, we can again see that here in blue, we have the inclusion change from wild type, and in orange, we have A1 change, and in green, SRSF1. So when we have a negative A1 score, we have, or a negative change from the wild type, we also have a positive increase in inclusion compared to the wild type. So these models actually work quite well both in these in vivo minigene studies and also in the in vitro studies in predicting the type of change that you would see given a certain mutation. As it happens, there's another mutation in the ACAT-M gene. This time it's located at position +7 in the intron 6, just after exon 6. So here we've indicated exon 6 in this light gray box, and here in red is the mutation. So that's in an A to G, and we thought that it might be TDP43. So we made this DeepCLIP TDP43 model based on multiple sources of CLIP experiments, both eCLIP, iCLIP, and ParCLIP, and got these binding motifs, which are look very similar to the already known binding preferences of TDP43. So we then made this binding profile across this 75 nucleotide input here, and again we see that when the binding profile, or when we have the mutation, then the binding profile predicts an increase in TDP43. So there is already a binding site here, and then this binding site is extended according to DeepCLIP. So again, we turn to a shorter. Again, we turn to a shorter oligonucleotide, so we could use that in an SPRI measurement. So here we also predicted with DeepCLIP, and we see the same type of binding profile here, that we have a predicted binding site, and then that binding site is enhanced and then elongated. We can also use DeepCLIP to just plot the difference to the reference sequence. So here is just the difference between the two curves. And that sometimes gives you a better impression of the effect of the mutation and which direction it's going. So here it's very clear that the mutation causes increased binding. So if we again take these two binding profiles and then do an SPRI measurement with recombinant TDP43, also called TADPP, we can again see that yes, we do have binding to the reference, the wild type sequence, but just as DeepCLIP predicted, we have an increase in binding. We then had, again, a mini-gene approach where we, again, ordered a mutation and so we had both the wild type and the variant. We then performed an SRI experiment where we knocked down the levels of TDP43. So here on the left, you can see that TDP43 is knocked down significantly. And then we transfected these cells and again performed RT-PCR and measured inclusion of the exon on an agarose scale. So here you can see on the wild type, there's no difference between knockdown and non-knockdown TDP43. But in the mutant, when we don't knock down TDP43, we have mostly skipping of exon 6. And then when we knock down TDP43, we have mostly inclusion. So this is demonstrate that TDP43 is actually involved in regulating this exon. If you look at the binding profile, you can see that there's also some motifs that are not affected or that don't really look like the UG-UG motif of the model, but have a fairly high profile. And that's perhaps context indicating, okay, so this type of context is contributing positively. So we have actually something that is contributing positively right downstream of the binding site. So the problem with this mutation is that there are approaches with using antisense oligonucleotides where you can produce a molecule that is complementary, so sort of an artificial nucleotide or RNA that binds to the site where TDP43 would bind, and then inhibits binding, and then that can remove the negative effect of TDP43. But in this case, the mutation is located so close to the normal splice site that if we do that, there is a very high risk that we affect normal splicing, so that would not be very therapeutically relevant. But we have this binding curve going up immediately downstream. So we designed these oligos to cover that portion instead. So we're not actually overlapping with the mutation and most of that binding site, but we are overlapping with this region where we have also a high binding profile. So if we perform an experiment where we treat the cells with SSO, either a control SSO or one of these SSO1 or SSO2, so splice switching oligo, we call them, we can see when we transfect with the control oligo and the wild type, nothing happens. That's the same for the SSO1 and SSO2. But in the mutant, here we have the control where we have predominant skipping just as before of the exon. When we treat with either of these SSO, we get predominant inclusion. So it seems like we are affecting binding and we're removing the negative effect of TDP43. So this is a case of where the binding profile actually is fairly essential to actually designing these therapeutics, which, because this is an intronic variation, the patient would, when treated with these oligos, would produce a completely normal functional enzyme and not have disease. So because these binding profiles, they sort of also show patterns of binding. And because TDP43 is this known factor involved in amyotrophic lateral sclerosis, ALS, where it's often observed to either be mutated itself and causing familial ALS, or it's depleted from the nucleus and sort of sequestered in the cytoplasm. And that way it's not able to exert its normal function in the nucleus. And TDP43 is known to repress pseudoexons. So as I mentioned before, pseudoexons are part of the introns that are not supposed to normally be included. And part of that is because they're repressed by splicing inhibitors. And one of those might be TDP43 in some cases. So losing TDP-43 from the nucleus could activate pseudoexons and be a part of what drives the ALS pathology. So there's been a number of studies. So here's one by Jiang and colleagues from 2017, where they were looking at pseudoexons that are specifically activated either in muscle or neuron by specifically knocking down TDP-43. So we took those pseudoexons and then we produced binding profiles across them, and then we took out this region 50 nucleotides upstream and 50 nucleotides downstream of the exon, and then 50 nucleotides into the exon for both the 3' and the 5' splice side. And then we produced these average binding profile plot here. And what's interesting is that you can see that there is actually a difference in how TDP-43 seems to bind in muscle-specific pseudoexons versus the neuron-specific. So in neurons, it's an almost equal distribution upstream and downstream. But in muscles, it's much more likely to be binding to the downstream region or the end of the exon. So because ALS is more affecting neurons than muscles themselves, this is important in looking at pseudoexons and which ones might be particularly important in neurons. We can also look at the heat map here, so this gives us for each of the pseudoexons the sort of the binding profile instead of as a curve. We're plotting out as a heat map with red as a high score and blue as a very low score. So again, here you can see that in the neuron specific, it's usually either or, and in the muscles there's so in some of the neurons there's actually very little chance of TDP-43. It's very blue here in the downstream region, whereas for the muscles it's almost always a high number of binding sites in the downstream region. So this is really an example of how binding profiles can provide these immediate patterns of binding without performing experiments. So just to summarize, with DeepCLIP, we have this architecture, this modeling of protein binding that incorporates arbitrary contexts. So it's not just the structure; it's just different types of contexts that are sort of for each model is specific to that particular protein, depending on the quality of training data. Of course, if you have bad quality training data, then models will also be comparatively worse. We also have these individual base scores, and as it turns out, that actually mitigates some of the biases that we see in training data. It also can inform this design of therapeutics because you get much more information about what is affecting binding of this protein, and we can use it to analyze these general patterns of binding. And because we have this binding profile, we're also better able to predict on variable length input. As you saw, this length of input, it does affect the binding score. Suppose you had only 10 nucleotides as input, that would be very different from having 100 nucleotides as an input. If you go across and look at the entire genome, and if you had or just 100,000 base pair, then having a score between 0 and 1 becomes quite meaningless. But we can actually produce these binding profiles across 100,000 base pairs, and then use a sliding window to identify possible binding sites. And we also make DeepCLIP available as a website, so you can use the already trained models and then get figures like I've shown you here with the binding profiles, and you can input both wild type and variant and get these difference plots where we have both the reference and the variant on the same plot. So it really makes it a lot easier for biologists to analyze if their mutations might be able to affect protein binding to that particular RNA. So finally, I just want to acknowledge and thank you all of our funders and all of the rest of the people in the RNA splicing and disease group, which is headed by Barbara Andresen, and in particular Alex, here he's circled in red. Alex and I designed DeepCLIP, and Alex implemented a lot of the neural network architecture and designed a lot of that. Also, Simon worked from Jen Baumbach's group, who's now in Hamburg, working on the DeepCLIP website. So, and I just want to thank you for your attention, and I'm happy to answer questions.
Thank you, Thomas, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, could this solution be used for sequencing alignment or sequencing comparison, for instance?
Okay, so thank you for the question. That's not something that we've considered much. I suppose if you're looking at cross-species comparisons and looking for conservation, not so much of the sequence, but of the regulation. So that could be binding of proteins to particular exons and regulation that might not appear immediately recognizable just from that sequence alone. Then using these protein binding models could give you some clues about more functional conservation than sequencing conservation. But we haven't really thought about using it for sequencing alignment.
Okay, thank you, Thomas. Our next question is, how long can my sequences be, or how short?
Yeah, so I've talked about this long prediction mode, and that's where we use this sliding window algorithm. And we trained all of these models with a maximum input size of 75 nucleotides and a minimum size of 12 nucleotides. So in the range of that, you will get this single, you will get the binding profile, you will get the single prediction score. But if you're using longer sequences than that, then you have to use this sliding window algorithm to produce a binding profile, and that does not produce an overall prediction scores because that becomes less and less meaningful. You can actually use very short input. You can even use one nucleotide as an input, and that will sometimes give you a surprising score, but of course it doesn't really make sense. So you still have to think about what does it make sense to use this type of this sequence input. So down to about 12 nucleotides is okay, probably, but usually we use around 15 to 20 nucleotides because we want to use also oligos to test, and then we use 75 maybe for a binding profile that covers this.
Okay, thank you, Thomas. And we have another question coming in. Do you plan to develop new models when CLIP data for other RBPs will be available? Do you train your model only in human data?
Yeah, so thank you. Yes, and absolutely. So right now we have about 195 different models on the website, and you can download the weights of that and also use them locally. So we have a local installation option for DeepCLIP in addition to the website. And those cover around 115 different proteins, and we are producing more models as they become available. So far, we've only produced human protein, but a lot of these factors are fairly conserved, so you might be able to get away with using them in different species, but we do plan to also produce models for other, probably mostly model organisms like mouse and rat and things like that. But you can always train your own models if you have a specific species that you're very interested in, and the data is available.
And Thomas, which is better to use, the score or the binding profile?
Yeah, so in my talk, I present a lot of data where we use this overall score to correlate with the data, the experimental data. But in many cases, the binding profile will give you that extra bit of information to actually, as a biologist, evaluate, okay, so does this actually seem to be affecting my protein? So a score is such a decrease in the amount of information that you get. So when you do look at the data, I think it's always important to look at that binding profile. But you can get away with the sort of prioritizing in some cases just using the overall score. So both have their benefits and negatives. The binding profile is more hands-on and it's more difficult to rank. If you have 100 different mutations, it's more difficult to rank them just looking at binding profile. Then the overall score is good to use.
Okay, fantastic. Thank you. We've got time for a few more questions. The next question is, Do you observe large differences between models for the same RBP trained on, e.g., PAR-CLIP and eCLIP data?
Yeah, so thanks. That is something that we do observe, and that's something that is sort of we provide on the website right now. We have different models where it's just trained on a single source, so it's either the PAR-CLIP or the eCLIP. Apart from that TDP43 model where we took a lot of data, and if you look at them, quite a few of them are actually quite different when you look at eCLIP versus PAR-CLIP, and then it's more of a question of how biased is the model. So you get slightly different motives and different profiles. So we are sort of planning to hopefully identify which models are the best and which CLIP method might produce the most consistently best performing models. But so far we're providing the models as they are and allowing you to sort of use them and then make your own decisions. But we do see differences between different CLIPs sources.
Okay, thank you, Thomas. We have time for one last question. Can I compare the scores of different models directly?
Yeah, so because they're trained independently of each other, their predictions are not like one-to-one, so you can't really compare them. But on the other hand, we do allow you to run multiple models on the same input at the same time on the website. So you do actually have that option of having a single mutation where you're comparing wild type and variant and then comparing those immediately. So you can sort of look at the binding profiles and see whether they're going up and down. But in terms of comparing absolute change in a numeric value, they're not directly comparable. But it's absolutely possible to look at binding profiles and score and see, okay, so is it going up or down in these two different proteins?
Thank you, Thomas. Do you have any final comments for our audience today?
I just want to thank you all for attending, and I also want to thank GenScript for sponsoring this talk and LabRoots for assisting. It's been very nice working with you and having this opportunity to present this work. And I hope you who attended the talk will try out DeepCLIP and give us some feedback. And I'm also available for questions via email if you have further questions. So thank you.
Thank you again, Thomas, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.